I'd like to begin today by first thanking the organizers for this wonderful opportunity to share some work that I'm really excited about, revolving around the ion channel TRPC5 as a novel therapeutic target in kidney disease. We find that there are many compelling reasons to work on kidney disease, chief among this the enormous and growing unmet clinical need in this space, especially as the rates of obesity and diabetes rise throughout the world, conditions that we know lead to kidney complications. And even though the kidney space has lagged behind others in terms of finding molecular targets for therapeutic intervention, we have gained significant insight into kidney biology by studying patient populations that harbor rare genetic mutants and present with kidney disease. Many of these mutations impinge upon a pathway that regulates the cytoskeleton of these cells and is characterized by the nodal signaling molecule RAC1. And through years of research that I won't have the time to go over through today, we have found that the ion channel TRPC5 is an important regulator of this RAC1 pathway. This is underscored by work in rodent models of kidney disease. So we see here in this top panel of images, immunofluorescence images that show us that TRPC5 is expressed in structures of the kidney called glomeruli that are delineated by this glomerular marker synaptopodin. So you see TRPC5 nicely colocalized with synaptopodin here. These glomeruli are comprised of a few different cell types, but chief among these is a population of cells called podocytes. And these cells are so called podocytes because they have these cytoplasmic extensions that are called foot processes. And you see these foot processes here in this electron microscopy images in these lower panels. So in a normal condition, this is what they look like. But when the kidney is exposed to an injurious stimulus, such as lipopolysaccharide here or protamine sulfate, we ablate these structures in a process called foot process effacement. And what's important is that when we genetically delete TRPC5, we are able to prevent this foot process effacement. So we're able to preserve the integrity of these podocytes. And that's shown here in the lower panels where KO refers to TRPC5 knockout mice. So what is TRPC5? Well, the gene TRPC5 is part of a superfamily of TRP channel encoding genes. And the protein product of the gene TRPC5 encodes this plasma membrane molecule that creates a pore in the membrane that will connect the external compartment to the cytoplasmic interior of the cell upon a certain stimulus. In this case, usually G protein coupled signaling. And for those of you who are GPCR nerds, in particular GQ11 signaling. Now, the key metric by which we usually measure ion channels is through the gold standard method of electrophysiology, where we record the electrical activity that is a result of channel opening and closure. And we usually express the relationship of current and voltage in the cell through these graphs, which we call current voltage curves. And what's important for the rest of the talk is that different channels have different current voltage curves. And so in many cases, we can really characterize a channel and know its identity based on the particular shape of the current voltage curve. And for TRPC5, this is a very characteristic W rectifying shape. And all that really means is that you have this curve downwards and this curve outwards. And that's all you really have to remember for the rest of the talk. So 3 years ago now, our group found a molecule that is able to inhibit TRPC5. So even though we showed that genetic deletion of TRPC5 is protective against kidney disease, obviously genetic deletion is not currently a viable therapeutic approach, which really led us to pursue small molecule inhibitors. And so we found this molecule called AC1903, which inhibits TRPC5 selectively over very related family members, TRPC4 and TRPC6. And you see here again those glomerular structures that I talked to you about. And so this is what it looks like in a normal wild type condition. If you overexpress the angiotensin receptor in the podocytes, you get kidney damage, as shown here by the cyst-like phenotype in this glomerulus. This is reversed or prevented by co-administering AC-1903 to these rats. Now, this is obviously a great proof of concept, but AC-1903 is not quite ready to be injected into a human, for example. It doesn't have all of the drug-like properties that we would need. In particular, we would prefer for it to be more potent as well as more stable. So in order to be able to find a compound that's based on AC-1903 but improves its properties, we really need to use the gold standard assay for ion channels, which is electrophysiology. But the traditional methods of electrophysiology are very laborious and time intensive. And so we wanted to create an automated assay that will ease this process. So just briefly here, what I'm showing you on the left is the traditional manual patch clamp method of electrophysiology, where you have a glass pipette that is pulled to a tapered end and then brought in close proximity to the cellular membrane, and at which point you will form a seal with the cellular membrane. But what we have at the Broad is this automated electrophysiology platform called the SynchroPatch that utilizes this 384 well plate format that has a glass bottom and has small holes drilled into the glass bottom in each well. So what this really allows us to do is to be able to record 384 cells in parallel, and greatly increases the throughput by which we can screen molecules for increased inhibition of TRPC5. So this is just a screenshot of what the instrument interface looks like. And what I'm showing you here is that I have been able to successfully develop an assay on this platform that looks at TRPC5 current. So what you see here in black is a baseline trace, so when the cells are unstimulated. But then we can add a known activator of TRPC5 called riluzole, and you see induction of these doubly rectifying currents that I described to you earlier. And importantly, we can then add compounds on top of riluzole and see whether or not they inhibit the generated TRPC5 current. And what we see for this specific compound is that indeed we do get inhibition. So here I'm just showing this in another way through what's called a diary plot. So tracking those current magnitudes over time. So this is baseline, and you see we add riluzole here and we get increases in magnitude, which is due to that doubly rectifying current. And then the same compound, when added upon on top of riluzole, leads to inhibition of the channel. And so what we can really do is now look at the percent inhibition, so comparing this epoch to this epoch, and in comparison to AC-1903, be able to really start to ask, do we get specific modifications that will allow us to increase the efficacy of TRPC5 inhibition. So this is just to show you that with a subset of compounds that we recorded both manually and on the SynchroPatch, that there is good qualitative agreement between the two platforms, which gives us confidence in the results that we will get from the SynchroPatch. So here in this graph, what I'm showing you is just a subset of the compounds that I've tested using this assay. And on the x-axis here, we are simply expressing inhibition relative to AC-1903. Which is shown by this dotted line. So what we're really interested in here is this tail of compounds on the right, which indicate compound efficacy, basically compounds that are much better at AC-1903 at inhibiting TRPC5 current, and for some of these, as much as threefold better. And we've published some of these results already last year. So then what we can really do is we can take those compounds that look very promising at single doses which was what the previous graph was showing, and test them over a range of concentrations to be able to construct what we call here as dose response curves, so that we can see over a range of different concentrations whether we really have improved the inhibition of TRPC5 current. And what we can see here is that a few of these compounds are left shifted with respect to AC1903, which is this heavy black marked line here. And what that means is that these compounds that are left shifted were able to inhibit TRPC5 at much lower concentrations. And so we think that this is very promising, and we're really starting to understand the modifications that we have to make to the AC1903 backbone in order to make it more effective at inhibiting TRPC5 current. So one of the remaining questions really in the field is whether or not the TRPC5 inhibition is relevant in humans. Because as you've seen from the previous slides, most of the work that's been done has been in rodent models or in heterologous expression systems. So we decided to really get at this question by leveraging new stem cell technologies. So Choa Kim and Mahesh, two talented scientists in the lab, took induced pluripotent stem cells, human-induced pluripotent stem cells, and derived podocytes from them using a podocyte differentiation protocol. So then I took these cells and I recorded from them using traditional manual patch clamp electrophysiology. And you see what that looks like in this image on the left. So this is my glass pipette, and I'm bringing it in proximity to this podocyte, which is in vitro resembles what we know podocytes look like. So they're these very large, flattened cells with these cytoplasmic extensions. And I can record current from these cells. So this is, again, just another current voltage curve, as I've shown you earlier. And then what I can do is add an activator of TRPC5, in this case, a compound known as englerin. And you can see this nice induction of that doubly rectifying current, and the shape, as I've described to you, gives us confidence that this is an ion channel of the identity that we care about, TRPC5. And to give us even further confidence in this, we can now add that TRPC5 inhibitor that I've described to you, AC1903, and indeed we do get inhibition of the englerin-induced current. So this now is, to our knowledge, the first time that TRPC5 channels have been shown to be functional in a human podocyte. And I just like to point out here that the early pharmaceutical work that generated AC1903 was invaluable to this research, because without the proper tools, then we wouldn't truly be able to dissect whether or not the currents that we're looking at are TRPC5 specific currents. So here I'm just showing you another current diary plot. So just tracking current magnitudes over time. You see this is baseline and this stem cell derived podocytes. You add englerin, we get that doubly rectifying current that is inhibited by application of AC1903. And when we stop application of AC1903, then we are able to restore those current magnitudes. And this is just statistical analysis looking at a population of these podocytes. So we are really confident now that human podocytes do actually express functional TRPC5 channels, which makes targeting TRPC5 a more viable therapeutic hypothesis. And this work has been submitted, and I won't have the time to go over this today, but we also show that AC1903 in this human-derived podocytes is able to prevent the damage caused by injurious stimuli. So what I've told you today is that we are able to screen through many different compounds to find inhibitors of TRPC5 current, which we know is protective against kidney disease. And we have proven that this TRPC5 channels are functionally expressed in human cells using the gold standard for ion channel work, which is manual electrophysiology. So with that, I'd like to thank the numerous mentors and collaborators by which none of this work would have been possible. in particular Anna Greca, who's the head of the Kidney Disease Initiative, our collaborators at the Stanley Center of the Broad Institute, and Corey Hopkins and his scientist Swagat Sharma at the University of Nebraska, who provided the compounds and have been great collaborators over the years. And with that, I'd like to thank you for listening today.